I would like to give the floor now to Viktória Feigl from the Department of Applied Biotechnology and Food Science. Dr. Feigl is an associate professor at this department, and she is the member of the research group of Environmental Microbiology and Biotechnology. Her main research interests include soil improvement, environmental microbiology and toxicology, waste reuse, and biohydrometallurgy. She also serves as a committee member of the BME for Sustainability Group and is the head of communication at the department and also another successful Horizon Europe participant. Viktória, the floor is yours.
Thank you very much. Yeah, again, a completely different topic that is coming, but I guess this shows the diversity here in BME. So I would like to talk about some of the research topics that are done in our research group. And this is basically 3 topics that I've brought you today. One is about using biochars for the improvement of degraded soils. The second is about cyclodextrin to alter microbiological communication. And the last is bioleaching of critical raw materials from bauxite residue. So let's talk about biochars that can be used for the improvement of degraded soils. I wonder if you know, but if you look at this map, what is marked with green are the soils that are more or less stable in the world, but all the others, which are marked with a certain color, these are somehow degraded. So we have some serious problems with our soil health. Like it can be the lack of nutrition, a decay in organic matter, or some bad mechanical stability, which after all affects our food production. So it's really something that we should deal with. And in the last 15, 20 years, our research group has been working with using various waste materials for the improvement of degraded soils. And one of our focus was on biochar. Yeah, So one of our focus was on biochars. Biochars are actually chars that are produced by pyrolysis from organic waste and waste-derived products. So that's how we get some waste-derived products that can be an alternative solution for improving our soils. And we had an international project with it was funded by the Norway grant, so it was a cooperation with Norway. And this one, we used various biochars to test so if they can be applied for the improvement of soils. Because the case is that we just cannot mix any kind of char into any kind of soils, but we have to pair them somehow. So it's we have to make some decisions based on the soil properties and the biochar properties to find the best options. So we created a multi-criteria decision support system for ranking and selecting the best chars from various soil degradation problems. And since that, we went a bit further, how to activate these biochars and how to produce better products, some kind of composite products. And we are currently running an EU-funded project, which was funded by the EGP Soil EU project. So this is a sub-project to this one. And we work in cooperation with Norway and Switzerland. And we not only use the biochars, but we try to make some composite products, which are using region-specific organic waste, and we compost it with the biochar. So, like for example, in Norway, which is not relevant. Oh, I don't know how. Yeah, okay. Yeah. Anyway, sorry. Which is not that relevant to us, but they have a lot of aquaculture waste streams. So they have to do something with this large amount of waste. It comes from fisheries. Or in Switzerland, they have a huge problem with dealing with sewage sludge or organic fraction of municipal solid waste. And this waste could be mixed with the biochar in a compost, and that can be used for specific soil problems. So we will try to make demonstrations at specific regions, which might be, soils might be, and and the climate is different in the north or in the Swiss mountains or here in Central Europe. So we try to prove that all these methodologies can work in these various regions around Europe. And my second topic today are cyclodextrins. I will explain all these because I know it's not the field of expertise of everyone. So to handle microbial communication to tackle some environmental and health problems. So first about microbial communication. I don't know if you have heard of this or not, but microbes can actually talk to each other. So they use signal molecules. So some emit some molecules, and the others can detect it and sense it and respond to it with other signal molecules. And this basically allows microbes to act as a multicellular organism, so they can organize themselves, like if they colonize a host or form a biofilm or produce virulence factors, or they decide to move somewhere or produce some exopolysaccharides, they can share this with each other, how to do it. And this is the way they basically adapt to changing environments. So there are a lot of processes that are regulated by this quorum sensing, and some of them actually might be problematic related to environmental issues or health issues, and also in agricultural and biotechnological processes, such as human infections or biofueling or even the spoilage of or food. So could we do something about this? The answer might be yes, so that we can somehow disrupt this communication between our microbes. And our research focuses on cyclodextrins. These are you can see it on the top. Yeah. So these are oligosaccharides, the special molecules that are hydrophilic inside, in the outside, so they dissolve in water, but they have a cavity where they can trap certain molecules. And for example, these signal molecules that are produced by the microbes can be trapped in these molecules. And this is called, again, like cyclodextrin-mediated quorum quenching. It's complicated, but the thing is that we can use these cyclodextrins to inhibit these processes regulated by the microbes. On the bottom figure, you can see one of our models. Actually, we use the bioluminescent bacteria. You know that there are some bacteria that can emit light. They usually live in the deep oceans in symbiosis with various organisms. And this light emission can be easily detected in our labs. So we can see if these molecules really work, because if this works, they stop the light emission, and this is something we can easily detect. But of course, it was just a model, and since then we moved to some more important things like biofilm formations. We worked with fungal and bacterial systems, and it was found that cyclodextrins can strongly inhibit biofilm formation. So this can be a solution to use superiority antibiofilm treatments. And this has a major importance in human health issues to fight against infections and proliferation of certain specific microorganisms. And just to mark that this research is done in cooperation with the Cyclolab company in Hungary. So the last topic is the bioleaching of critical raw materials from bauxite residue. I guess also bauxite residue might be familiar to you. It's also called red mud. It's the waste of aluminum production, which is aluminum oxide, and it's estimated that there is more than 400 billion tons, 4 billion tons deposited worldwide. It's highly alkaline, saline, might contain some toxic elements, and has a very small particle size, which makes this waste material really problematic regarding storage. As you also might remember that in 2010 it caused a major catastrophe in Hungary when there was this red mud spill in Ica. So it would be nice not to store this material, but to use it for something. And there were some attempts using it as a soil component or also in the construction industry. But we are focusing now is to use them as a source of critical raw materials. Critical raw materials are elements that have high economic importance. So, for example, in our LCD screens, we have some, or also the wind mine turbines, or in our electric cars, the accumulators. But in the EU, we have a strong dependence on their import, mainly from China, but also other parts of the world. So it would be good to have sources within Europe for these critical raw materials. And what has been developed in the recent years is the biohydrometallurgy. So some hydrometallurgical and pyrometallurgical processes has been developed. It's more under piloting and not at the large scale. But what we offer or what we try to work on is using microorganisms to extract these elements from the bauxite residue, as they also, some organisms like the fungi, some fungi that you can see on the bottom, they also both produce acids and some complexing agents, and so that these important elements can be released from the solid matrix, and we can use them further. So these biotechnologies may offer an alternative to the above-mentioned processes, like it can work at lower temperatures, not like pyrohydrometallurgy, which uses several hundred degrees Celsius, or we don't need harsh chemicals, like in hydrometallurgy, where they use very strong acids, hydrochloric acid, sulfuric acids to get these elements. And we also have the biomass as a byproduct. And we've been working on a project that's been funded by the National Research, Development and Innovation Office, and we try to use some fungi to extract various elements from the bauxite residue, and we were successful in getting aluminium, scandium, gallium, titanium, vanadium out of the bauxite residue, even at very low temperatures. So it works at room temperature. We don't have to use elevated temperatures. And we also work in cooperation, again, to use some waste materials as a carbon source, like from sugar companies, sugar producing companies, or also the beer production has some waste that can be used. And we are also working with actually the biggest alumina and aluminum producing company with Greece, who are also interested in such biotechnological processes. And as Borbala mentioned, I also wanted to showcase this, that we've been participating in two Age 2020 projects. And as I remember you said, it's also good to start with something small. And actually, in this project, we didn't have such a huge roles, but we did ecotoxicology testing. So ecotoxicity testing is using these small laboratory plants, animal microbes to predict the potential toxic effect of chemicals or waste for the environment. So in the first project, we followed up the development of scandium production process, where we examined the potential toxicity of chemicals used in the processes and also the waste in the end. And in the second one, we followed up some bioremediation processes where we try to prove the success of this remediation methodologies also by toxicological tests. And we don't have that huge horizon projects right now, but we still work in international cooperation with some partners, like we also participate in other projects from our faculty, a coordination and support action, the Beaming project, which deals with bioeconomy excellence, or we also participate in a Marie Sklodowska-Curie Action Staff Exchange project, the Clean Water, where the focus is on plasma technologies for water treatment. And we also have a bilateral project with Croatia, which is, again, another topic, investigating the effect of microplastics in soils for soil-dwelling organisms. So that's all, and thank you for your attention.